With this, I would now like to go on and show you how we actually perform the fully automated determination of phosphatidylethanol with our system. In order to do that, you need a autosampler for DBS. Then we use 2 binary LC-MS pumps, a mass spectrometer, which is, for example, an Shimatsu 8050 or a Sykes 5500, so a quite sensitive one is needed, and furthermore a switching valve, otherwise you are not able to perform online SPE LC-MS, as I will show during the next few slides. The detailed instrumental setup we are using is depicted here. We use a trapping column, which is depicted here in orange. This is a Synergy Polar 2 centimeter reverse phase column, which is fitted within the DBS instrument at the 10-port valve instead of a sample loop. And as analytical column, we use a lunar reverse phase C5 column with a length of 5 centimeter. Afterwards, sample preparation takes place, whereby we load or we elute the DBS spot onto the trapping column. Everything not interacting with this trapping column is directed to the waste. Afterwards, we further flush the trapping column a bit and then direct the flow onto the analytical column by flushing the column backwards. And onto the analytical column, once the compounds are there, separation takes place. And while this separation takes place, we already wash here the extraction head and can clean the trap and load the next sample onto the trap. This means that we have sample preparation of 2 minutes, LC-MS run of 5 minutes, and by overlapping, we have a total runtime of about 5 minutes for the sample analysis. A bit more in detail, we can see that here. On the left, we see the trapping column, which gets preconditioned with formic acid and water. Afterwards, we load the sample onto the column and start at time point 0 by flushing a little bit more with water, then changing to a more organic composition, which is 70-30 acetonitrile water with some ammonium acetate within. And once phosphatidylethanol is eluting from this column, we switch, indicated by these yellow bars, onto the analytical column in order to transfer the analytes. The analytical column was preconditioned with a very similar solution, and afterwards we perform gradient elution by adding some isopropanol to separate the 2 compounds, PEth-16O-18-2 and PEth-16O-18-1. How this then looks as a chromatogram is depicted here on the top right. We included both internal standard PEth-16O-18-1 D5 and PEth-16O-18-2 D5 to get our method more accurate. These compounds were produced in-house out of phosphatidyl-choline reaction. The range of the method is from 20 to 1500 nanograms per milliliter based on a 6-point calibration on a normal basis. We include 3 quality control samples at concentration of 20, 45, and 1180 nanograms per milliliter. The extraction itself is taking place with 40 microliters onto the trapping column. The detection of the compounds is performed in multiple reaction monitoring mode. We fully validated the method, investigating the linearity, accuracy, and precision, and everything was within FDA guidelines. Considering the linearity, we were at least at 0.998, and we had a for PEth-16O-18-1 and 0.997 for PEth-16O-18-2, and we had an extended linear range up to 2,500 nanograms per milliliter with a correlation coefficient of at least 0.99. The LOQ was confirmed at 20 nanograms per milliliter, the LOD at 10 nanograms per milliliter, which is ideally suited in order to perform PEth analysis, as you usually apply a cutoff of 20 nanograms per milliliter to distinguish between abstinence and moderate alcohol consumption. On the right, you can see how the signal intensity looks based on the standard measurement for PEth 69018-1 in the calibrators of the blood. What we afterwards did was investigated how good our method actually is by performing a comparison between whole blood extraction versus our fully automated DBS analysis approach. And as you can see in the middle, there's very high agreement between those two methods. However, if you use the fully automated DBS analysis approach, you have much less steps which you need to do. You just have to add the card basically into the autosampler. You have to build a batch and you have to hit play. Whereby, on the other hand, for our manual whole blood analysis, what we did was, first of all, labeling all the extraction containers once we had the labels prepared, then pipetting the blood samples into Eppendorf tubes, adding extraction solvent and subsequently internal standard, shaking the whole samples for at least 20 minutes, then centrifuging for 10 minutes, afterwards transferring the supernatant into a glass vial, evaporating this whole solvent, and afterwards reconstituting the sample in order to be ready for the LC-MS injection. We afterwards, due to the fact that we got very promising results here, went on and compared the manual whole spot extraction to our fully automated DBS analysis approach, which is just a sub-punch extraction. And also here we got very high and good agreement. However, with the fully automated DBS analysis in comparison, you just need 5 minutes instead of several hours in order to prepare all the samples, and then still have the 5 minutes of analysis to determine the concentration of your sample. With this, I'm already at the end almost of this webinar. If everything of this PEth stuff was too complicated for you, upon request, CAMAG decided to provide the complete PEth package. This means that we can provide customized DBS cards with individual printing in collaboration with our partner Ostrom. We can provide you with PEth reference solutions, with protocols for the production of your calibrator and QC samples, protocols for the production of internal standard solutions, and furthermore, standard operation procedures for the sampling of, for example, capillary DBS samples. We can provide you with column material and cartridges, connections and tubings, and furthermore, the method installation and optimization, so that also you can do fully automated DBS extraction. To summarize, what I just presented was a fully automated method for the DBS PEth analysis. The presented analysis allows you to determine phosphatidylethanol 69018-1 and 69018-2 with a total analysis time of about 5 minutes. The setup here is ideally suited for the high throughput analysis of phosphatidylethanol. Within the machine, you can load 500 cards, which can be up to 2,000 samples. The analysis of both major PEth metabolites increases the confidence of your result. So it's much better to include also 69018-2 and not just go for the 69018-1. And as already mentioned, if you have barcoded sampling cards, you can integrate the process into your laboratory information system. So basically, you would not have to actually build a batch if you got your right setup within the laboratory where you can recognize each sample just by the barcode within the analytical run by having the camera system set up properly. With this, I'm at the end of this presentation, and I'm happy to answer your questions. If you have more detailed questions or requests, please send me an email, and I hope you enjoyed the presentation. Thank you very much for your attention.